Hello everyone. In the last few classes, we learned that how do we fetch information from the DNA sequences. The next logical step could be to perform experiment which are guided through those sequences. For example, if you have predicted a gene, you would like to study that gene further. So you could either do expression profiling, gene expression. You could also do cloning experiment. Right. So in these experiments, you try to study whether your prediction was correct and the gene is expressed. If it is expressed, in what conditions the gene is expressed, right? And you can also do a cloning experiment where you clone the gene of your interest that you have predicted into E. coli and express the gene at a very high level, and then purify the protein and study its function and structure and other things. Now, when you do these experiments, one of the things that uh, which is required is to perform an experiment called PCR. Now, this is a molecular microscope kind of thing, polymerase chain reaction where you amplify a chunk of DNA. All right, so if you have a DNA sequence, okay, coming from a cell, you somehow purify this and you want to amplify, you want to make molecular copies of these, a lot of copies, okay, and that can be done using PCR experiment. Now, those who are not having a background in biology or molecular biology can get back to PCR uh, in books, and you will learn that it's a rather straightforward uh, concept. It uses the concept of DNA replication. So basically, you start the DNA replication in convergent manner from two ends, and then you are able to amplify. So you start from here, and you start from here, and then you are able to amplify in between region, right? So, and I also believe that this must have been taught to you in first year and second year biology classes. So what exactly do we do in a PCR experiment? So you have your Eppendorf tube, let's say. This is your Eppendorf tube, right? And you have all your solutions inside. So what all do you have? So of course you will have water inside. You have a buffer to maintain the pH. You will have ions, positively charged ion like magnesium or sodium. Uh, you will also have your DNTPs, which are required. So DNTPs are incorporated during your polymerase chain reaction, replication reaction. You will of course need a polymerase enzyme, and in this case we use a stable, thermostable Taq polymerase, right? Taq polymerase. And you of course need your source DNA that you want to amplify. So one of the important thing that we need in these in PCR experiment, and that's what we are going to talk about today. We need to start the experiment. We need to start the PCR through some end, right? So in this case, we are taking these two ends. So we need some starting DNA material to anneal to the ends, right? And those are known as primers, right? We need primer sequences. So this is forward primer, this is reverse primer. So you'll have a pair of primer, right? Basically a pair of primer. How exactly you select the primer sequences is a computational problem. So what we are going to do today is we will learn something called primer designing. Okay, now designing is a misleading term here because we are actually not designing anything. We are not making certain combination of nucleotides to get these primers. We are actually selecting primers. Okay, these sequences are present, complementary sequences here are present, and we are just selecting them in a manner that we find appropriate combination of primers. This is actually primer selection, truly speaking. So let us see how do we do this. Given a sequence, how do we select a best pair of primers for my PCR reaction? Okay, so this is my primer selection. Primer selection. You can call it a primer design, but don't get misled by the term designing. We are not designing anything. We are selecting an appropriate pair of primer. Okay, so let us assume that we have this target DNA, okay, my source DNA that I want to amplify. Now you can define, you can decide about how much length you want to amplify. Do you want to amplify a small portion of this or a whole chunk of this, or what is appropriate for your experiment? For example, if you're doing expression profiling through RT-PCR, 100 base pair amplicon, 100 base pair of the product size to be amplified is sufficient to measure the expression level of a gene, right? Now, once we have this source sequence, the first thing that you need to do is to define from where your forward primer should be selected and from where the reverse primer should be selected. So let us say that you have defined this range here. For the forward primer, this is the range from where the forward primer should be selected. And likewise, you defined a range for the reverse primer. That my reverse primer should be somewhere here. Right? So now the objective is to, and the length of the primers are generally 20 base pair, 18 to 22 base pair. That's the appropriate length of a primer. So you can have this as one primer and this as another primer, some sequence complementary to this region of the sequence. Now there is no reason to choose this immediately without testing whether this is appropriate or not. This might be appropriate, might not be. Right? So there could be another sequence somewhere here and somewhere here, which is making a correct pair. And likewise, you will have a lot of possibilities of primers to be selected. Okay, there will be a lot of sliding windows over here that can be selected as your primer, right? Likewise, your reverse primer. Okay, and it turns out that one of these primers is going to uh, it will be better than others, right? So let us say that uh, somewhere here, this primer, this forward primer, and this reverse primer were very good choice. Now, and this choice is, so how do we make this choice? That's what the primer selection is. So if you have all these combinations of forward and reverse primer, so these are your forward primer here and these are your reverse primer. You make all possible combination and start scoring each pair, right? So when you score each pair, you are looking at certain properties of the sequences of forward and reverse primers, okay? which makes them good or bad. So we have to score them. So the idea is that we need to score them or penalize them in score or the penalty. Okay, so if score is high, the penalty should be lower, right? So either you score them and then you look at the maximal score, or you penalize them based on certain properties and say that I will choose the pair which is having the least penalty score. So this is how we select the best primer pair. So let us now look at how exactly we do this. So it is clear that we need to look for certain parameters, right? So we have to score them. So that scoring should be done based on certain parameters. So let us write parameters here. What are the parameters that we bother about? Of course, the length is one parameter, but it's one of the trivial parameter. Whether the length is 18 or 22, it doesn't make much difference. So the real parameters would be following. One of the parameter that we can look into is the GC content, right? Now, why GC content? Because if your primer pair is low at GC content, there will be problem with the annealing. You need 50 to 60% of GC content in the primer sequence to have appropriate annealing, right? So we also score based on GC content. If there is a primer sequence which is having 10% GC content, it's a bad primer. So we should make sure that there is sufficient GC in the sequence. We need to look at the melting temperature of the primer, okay, which should match with your PCR setting. When you set the PCR, there is a one step where you melt the DNA, the duplexes, uh, around 90 to 100 degree centigrade. And then there is an annealing temperature. So TM actually makes sense during annealing, right? So there shouldn't be more than uh, 5-degree difference from the annealing temperature. So we need to look at what is the melting temperature of primer sequences. So we will see how do we calculate it later on. And then we need certain other important scores. For example, self-annealing score. In a moment, we will talk about self-annealing. What is self-annealing? Basically, primers can anneal to itself, right? So then they will make a primer dimer and we will learn about it. Self and annealing, the 3' end of the primers are annealing to each other. So this is self-annealing, self-annealing is for the same primer, either forward or the reverse. You can also have a scenario where pair of primers are annealing to each other, forward and reverse pair, and likewise their end is also annealing, which is pair and annealing. We will see how pair and annealing is slightly different from pair annealing. And likewise your sequence, your primer sequence can actually mishybridize to non-target, non-specific target in the genome or in your DNA sample. So when you purify your DNA, the whole DNA is there, and it may hybridize to non-specific sites. So that's called mispriming annealing and mispriming and annealing. So these are the main parameters on which we are going to score our primer sequences. Right. So now how do we score it? So we will look into these parameters, but let us first look at the scheme. What are we going to do after scoring them? Okay, first of all, we need to have a reference range, right? What is the ideal value that we want for GC content, TM, SA, SEA, and so on? So let us say the reference values, right? So the reference value would be, let us say for GC content, I have defined the reference value of 55%. and it should have 55% of GC content. TM, I have decided to keep it at 65 degrees centigrade. Now SA, I would say I don't want any self-annealing. So my ideal reference value for SA should be 0 unit, right? And likewise, it should be 0 self-annealing, pair end annealing, mispriming annealing, and so on, right? And likewise, you can also have a length parameter. So since the length parameter seems trivial here, I haven't mentioned it. Now, the second thing is you need to calculate. So these are the parameters that you want in ideal scenario. And now you need to find out for one pair at a time, what is the observed value, right? So I'm going to calculate the observed values for one pair. So remember, we have multiple pairs. If you look at here, you need to make all possible combinations of forward and reverse. So let us say I took this pair that I have highlighted in red, and then I want to score it. So I'm going to look at each sequence individually. And then I say, let's say my GC content was 45% for one of the primer, 45%. My TM was 62 there, right? And this score was 10 over here, 5 over here, 8, 4, uh, 5, and 2. Let's say these are some arbitrary score, and I will show you how do we actually calculate these scores. And the first thing is, now you have two vectors, right? Now you need to find out, get the difference vector of this, right? So you need to get the difference vector. So the difference would be following. You will have the difference here. This is going to be 10% here, right? This is going to be 3 degrees centigrade. This is 10 unit. This is 5 unit, 8 unit, 4 unit, 5 unit, and 2 unit. Right? Now, I can't just add up all this and say this is my final score, right? Because GC content and TM are having different unit. One is the percentage, other is centigrade, Celsius. And these units are, I'll show you how do we calculate these values. So they have separate unit than what we have over here. So basically you need to have a weighted sum, or in other ways, you define your penalty scheme in a way that it is weighted. You give certain weightage to certain difference and less weightage to others. So what we do is the following. So I'll say penalty here. I'm going to take penalty. Okay, rather than scoring, I will penalize something. So here is the scheme. This is important now. So for every 10% change in GC content, I would say +1 penalty, +1 for 10% change from the ideal value. And likewise, now this will be different here. I will say +1 for every 1 degree change. Okay. So you see, I'm giving more importance to TM just because even 1 degree change in TM is important, right? It's important biophysical property. While the GC content, we can have in percentage, we are a little bit flexible out there. So these are all educated values from the literature, from the past experience. People learn that which parameter is more important in a PCR reaction. And likewise, this is also optimized based on a lot of hit and trials. And it turns out that people say, I will give +1 penalty for every 10 unit change, for every 10 unit change. And +1 here penalty for every 5 unit change of SEA. And likewise, okay, +1 here for every 10 unit change of pair annealing, 1 here for every 5-unit change of pair end annealing, plus 1 here for every 10 unit of MA, plus 1 for every 5 unit of MEA. Right. So let us score for this particular primer that we found the difference for. Right. So what will be the score over here according to this schema? So I'll give a penalty here. So that's 1. So I have plus 1 here, right? This should be plus 3 here. This is plus 1. This is plus 1. This is plus 0, plus 0, plus 0, plus 0. So my total score is 3, 4, 5, and 6. So my total score is 6. That's my penalty. And lower this penalty is, better it is. So 6 seems to be a reasonable score. So this primer pair seems to be a good one. So any score less than 10, any score less than 10 seems to be good one. But then you need to look at what is lowest, even below 10, what is the lowest score, right? So this is a scheme by which we calculate uh, the best primer pair among all possible pairs. Okay, so now let us look at how do we actually calculate these parameters, right? What is the method to calculate these parameters? So let us first look at TM. Now, first of all, there is an arbitrary method for calculating TM. There is an arbitrary method. And some of you might be aware of, and that method is, it says that you need to look at GC and AT content and say my TM is actually 4 times GC content plus 2 times AT content. Now this equation is fine as far as the short primer sequences are concerned. Why? Because for shorter one, let's say I have 20 base pair, and this will be the unit will be your degree centigrade. For example, for 20 base pair, if I have 60% GC content, which means 12 bases are G or C, so 4 times 12 plus 2 times 8, right? So this is going to be 48 plus 16. This is around 62 degrees centigrade. This is fine. This roughly works, right? The problem is for the longer sequences, isn't it? So for example, if my sequence is longer, then this is, let's say 100 base pairs with 60% GC content and 40% AT content, then 4 times 60 plus 2 times 40 is 240 here plus 80 here is 320 degrees centigrade, right? So this is not correct. For the longer sequences, it doesn't work. For longer sequences, this method doesn't work. This is the arbitrary method. So there is another method which is the thermodynamic one, and many of you might be aware of that. I think this is given by Breslau. Somewhat, this is spelling roughly. Breslau et al., the author. Okay, so how does this work? So the TM is given by your change in enthalpy in your renaturation statistics, renaturation dynamics of the DNA denaturation dynamics uh, of the DNA duplex, right? and then divided by change in entropy plus the gas constant log of C by 4, where C is the concentration of the DNA, minus 273, which is the correction for from Kelvin to Celsius change, plus 16.6 times the ion concentration. Okay, so this is in degree centigrade again now. Now you might wonder that, okay, this equation is out there, then we are not going to do experiment here. We are given a set of sequences for which we need to calculate TM scores, right? So I don't want to do experiment to calculate delta H and delta S. So it turns out the values of delta H and delta S are already given in the literatures. For example, not for the whole sequences, we will have the values for each dimer. Okay, for example, you can have a dimer AA, which is A followed by A. So this is ith position, I plus 1 position, or TT, right? Likewise, you will have AT, GC, CC, right? And AT, CG. For each of these dimers, right? Delta H value and delta S values are known. So these values are known for each of these dimers, right? And of course, these are coming from experiments. So now we have for each dimer the delta H, delta S value. You can actually calculate for a given sequence. For example, now your sequence is following. This is your sequence. What you do, you take the window of 2 is you take this timer. So let's say this was your primer till here to here. This is the primer sequence. You take the window of 2 base pair sliding windows, right? Right, till the end of the sequence, and for each one of them, you already have the delta H and delta S, right? So what you do, you sum them up. So your equation, this equation looks like following now. You are basically having sum of delta H uh, from from first position to nth position. N is the length of the primer, length, right? Delta H from ith to I plus 1 position. So this is one dimer. I to I plus 1 is one dimer, right? And likewise, you do summation over here for delta S. Delta S from I to, so this is I here. I to I plus 1. This is dimer, and the delta S for one dimer, and then sum of the all the dimers, sum of the all the sliding windows over here. Right? And the rest of the equation remains same. And likewise, right? So if you have this table available with you, and these are available, okay, you can actually calculate the TM score, thermodynamic TM. So this is the way we calculate the TM of each primer. Okay, so now let us look at how do we calculate SA and SEA. So we perhaps need to draw a sequence over there. So SA and SEA. So we only need to learn about SA and SEA. Then we can extrapolate the same thing to PA, PEA, and MA, MEA. Right? So let's try to draw a sequence over here. Okay, so let us say there is a primer sequence which looks like this: G, C, P, T, A, T, A, T, G, C, C, T, Right now, this is your 5' end, your 3' end. Now, this is not the lone sequence in your tube in which you are performing your PCR experiment. There will be a lot of this. So you take a very high concentration of primers. There will be a lot of these primer copies of these primers available in the same tube. So there is no reason to assume that they will not interact and cross hybridize with each other. So if the same sequence comes in inverse orientation, it is going, and if there is sufficient complementarity to this sequence, then it can hybridize to itself. For example, if I draw it in inverse direction and I see some homology, I'm just looking at one orientation. Okay. So C, G, G. I'm now typing this sequence from this end to the other end, inverse direction. So this becomes T here, A, T, T again, right? And then C, C, G, right? And then again T, A, T, A, T, T, C, G, right? So this is my 5' end now, and this is my 3' end. Okay, now how do we score? If you have this orientation, then there might be some complementarity and the hydrogen bonds can be formed, right? So for example, there will be hydrogen bond over double hydrogen bonds over here, right? So you can write, so, okay, so I'm just typing the complementarity over here, just marking the complementarity. T is not complementary to T, A is complementary, and then this is also complementary. These three are also complementary. So this is one orientation, right? We will see that, you know, what do we do with multiple orientations. Now let us see how do we score it. Now for every hydrogen bond between C and G, we give a penalty of +2. Okay. So here this is going to be +2, +2, +2. And for every A and T, I say +1, +1, +2, +2, +2. So this is going to be my SA score. My SA score is just the sum of these. So this is 6 + 6, 12. This is 14 is the score, right? 4, 6, 7, 8, 9, 10, 11, 12, 13, 14. This 14 is my SA score. Okay, this is the way we score SA. Now, what is SEA given this orientation? Now, SEA is for the 3' end. Okay. Now, what is the complementarity right at the 3' end? The continuous complementarity in the 3' end. For example, this is the 3' end, and there are 3 in a stretch which are having complementarity for this particular hybridization, right? C to G, G to C, G to C, and then there is a gap. So the moment this continuity is broken, I don't consider any further. So I need to consider only these 3, and likewise these 3, right? It's the same thing basically. If you're scoring this one, this is getting scored automatically, right? Now, so the score is actually 6, right? It's just 6. So my SEA score is just 6. You don't have to actually repeat it for both of them. Okay, now you might wonder that what is the need of looking at 3' end and not the 5' end? Here is the need. For example, you know that the DNA replication as well as transcription, it happens, the amplification of DNA, the DNA synthesis particularly happens from 5' to 3' direction, right? Now what happens, let us say the DNA polymerase came and it recognized this end, okay, 3' end, and it found a template. Now, important thing is that the DNA polymerase will find a template to be amplified. right? So it will start making DNA on this direction till here, right? So it's actually amplifying the wrong product. It is amplifying the primer itself. And likewise, it will amplify this one till here, right? Because there's the availability of template. Now, if you invert it, for example, you can think of a scenario where sequences are hybridized like this, where 5' end is actually having some complementarity. Now, you might say that it can recognize here, but the synthesis doesn't happen in this direction, right? It only happens 3' to, sorry, 5' to 3'. So it happens from 5' to 3'. So 3' is this way. So it happens in this way. This way, there's no DNA synthesis. So this 5' end annealing is irrelevant. So this is why we look for 3' annealing. Now, which means that now primer is amplifying itself. So they make something called dimers. This is called primer dimer. Okay, primer dimer, which are bad. So they will keep on amplifying themselves, and you will see a thick band at the bottom of your gel. Okay, if you run the gel electrophoresis, you will see a very thick band at the bottom, which is the primer dimer thing. This is bad for you. It means your PCR was not optimal one. It's actually most of the resources were used in making primer dimer rather than amplifying your product of interest. Okay, now the question is following. If this is the case, then how do we decide upon to which orientation to look at? For example, you can have so you can have this orientation of 2 primers, right? And then you can have all possible orientation where uh, the primers are hybridized in different ways, right? In different orientation. So you will slide these 2 primers over each other through its lengths, right? So throughout the length of the primer, you can have different combinations by sliding over these 2 primers over each other. So you will get different combination like this. So, so how do you decide which one to be used? So you look for the worst score, which means that the max score, max SA, let's say over I, and SEA, right? I is the index of each of these combination. Right? There could be n different combination. So we take max of this, which means that I'm considering the worst case scenario. In worst case, what is the primer dimer formation tendency? Worst case scenario. And that will be my SA and SEA. So this is the way we calculate the SA and SEA. Now you can extrapolate the same thing to PA and PEA, right? So in PA and PEA, the only difference is that now you are looking at one of the primer would be your forward primer, and the other primer is different one now, but they can still be complementarity. This is the reverse primer. There can still be some complementarity, right? Isn't it? There can still be some, so you have to score these also for, and those scores will be now known as PA and PEA. And again, you look for the max of these scores. In very similar manner, you will have, you can score the MA and MEA. Now, we don't have 2 dimers now. MA means mispriming annealing, so which means that, or mispriming simply. So let us say you have, this is your target where your primer should be binding here, forward one and reverse one. So of course, you will have 100% complementarity over here. But there is always a possibility that in your tube you had other DNA sequences, right? So there is always a possibility that the same primer, this one, is actually finding some complementarity somewhere here, let us say, right? Isn't it? And somewhere here and so on. So there can be a scenario where, so you have to score this now, right? What is the worst case scenario here? Now, there can be a scenario which looks like this. This is your 3' end, right? And there is a very good complementarity at the 3' end of the primer. Now the 5' end is opened up because there is no complementarity here, so it's not hybridizing to the non-specific DNA. This was your specific DNA target. This is non-specific, right? However, the 3' end is annealing there, and now again your polymerase will come, recognize this end, and this is your template, and start amplifying this, right? The wrong product, isn't it? So which means that you need to score these also. So once you score this scenario, then you will be able to take into account the mispriming into non-specific target. Okay, and the scoring is done in same way. So this will be your MEA score here, isn't it? And likewise, you find out the worst case scenario here again. You find the max penalty. So this is the way you compute self-annealing, paired annealing, and mispriming of the primers. All right, now when I mentioned about the method, this method, this might be fine if you have limited region here to look for primers. So then you will have limited number of forward and reverse primers. So the possible combination of these primers would be rather limited, right? But there can still be a scenario where this scope is actually increased dramatically. And there can be multiple different reasons why that scope is greater. For example, there's a long sequence over here, and now you need to find out your forward primer from this region and reverse from this region. So now you will have lot of forward primers, right? All different sliding windows over here and so on. And likewise, lot of reverse primers and so on. And you can also have a scenario where you're not using two primers. You're actually doing multiplex PCR where you have multiple products to be amplified. A lot of assays in molecular biology where you use multiple pairs of primers in the same reaction. Then you need to look for different kind of cross hybridization among the primers themselves. So your computation of making all different combinations of these two increases dramatically if your range from which you're selecting forward and reverse primer is increased, right? Then how do you actually get the best primer pair? So you actually don't get the best. What you do, you find the optimal primer pair. So this, which means that you need to uh, do the optimization rather than having a deterministic solution. So you heuristically do your calculations using an optimization algorithm. There are a number of optimization algorithm which includes the genetic algorithm, simulated annealing, steeped descent, conjugate gradient, and so on. So again, you can try any of those to find the optimal solution to your problem. If the search space for primers is increased dramatically and there is no real-time solution available, right? Now, let us look at some little bit of biology of primer designing. Now think of, now this is also your exercise, one of the assignment. For example, think of a scenario where uh, you are looking at the eukaryotic gene, right? So eukaryotic gene will have exon and then intron, long intron in fact. Right, so this is your DNA, right? 5' to 3'. This is your genomic DNA. Now, when you do expression profiling, you're not interested in genomic DNA. What you do is the following. You uh, actually isolate from yourself the RNA, mature RNA, right? So that would look like following. It will not have introns because splicing has been done by the cells. First exon concatenated to second one, to the third one, and to the fourth one, right? So this is my exon 1, exon 2, exon 3, exon 4. Now they are together, right? This was my intron 1, intron 2, intron 3. So this is what we got after transcription, right? Transcription plus splicing. Okay. Now, what we do here is we actually try to get our cDNA. We make cDNA from this because all the PCR reactions actually work on cDNA. So we make a cDNA of this. This cDNA. So the only difference between RNA and cDNA is that you have, this is DNA first of all, you have thymine instead of uracil, and then this is double stranded, this is single stranded RNA, this is cDNA now, right? This is double stranded DNA on which you can perform the PCR. So this was your mRNA here, your cDNA. You perform the reverse transcription reaction using reverse transcriptase and you get this. Now the question is, now you were supposed to do PCR of the cDNA to find out how many copies were there, which will indirectly tell you how much mRNA was. So you need to select primers here. So from where will you select the primers? So let us say that as I mentioned earlier, 100 base pair amplicon is sufficient to do expression profiling, and you will say that I find a good primer pair over here. Now this is going to amplify this region, right, of the transcript, and you measure that using quantitative methods like real-time PCR and so on, and say that what was the expression level, compare it to the some control conditions, or compare with the housekeeping genes that how much this gene was expressed, this particular gene was expressed. Now let me change the scenario slightly for you guys. Let's say that in my tube where I had this cDNA, I also have contamination of genomic DNA, which can happen when you purified your mRNA here. This mRNA also had some contamination of genomic DNA, and this is the routine thing, it does happen, right? So if this is the case, then what will happen when you perform the PCR? So the same primers which are binding to cDNA in the exon 2, this is your exon 2, right, can also bind to genomic DNA over here. The same primer pair can actually hybridize to genomic DNA and start amplifying genomic DNA, which we don't want. Why? Because we are looking at how much this gene is expressed. So mRNA, which is reflected in cDNA, the copies of cDNA, right? And if I started amplifying genomic DNA also, then my expression profiling will be erroneous. There will be a bug in my readings. So how should I choose my primers that I get rid of genomic DNA? Now, some of you might also say that when I was purifying mRNA, then then I can add an enzyme called DNase, which will chop off the DNA but only keep the RNA. So then I will don't have genomic DNA contamination in my cDNA sample. Well, let us say that I don't have DNase enzyme, or I want to do it in a cheaper manner, that I don't use DNase, and I design the primers in a way that it is going to amplify only the cDNA and not the genomic DNA. How can we do that? So one of the ways people do is uh, they will design primers known as intron spanning, which means that instead of amplifying exon, I will choose primers in a way which will span one intron or one of the exon-exon junction. Let's say I chose one primer from here and another primer from here. So now this region over here, this region is basically what? Your exon-exon junction, right? Now this exon-exon junction was actually having intron, isn't it, in your genomic DNA? So this is why we call this set of primer which are spanning exon-exon junction as intron spanning primers. Now more often the introns are very long. So if this product here was just 100 base pair, when we consider the cDNA, the same primers when they bind over here, so the same primer can again bind to genomic DNA, no doubt about that. So one will bind here, another will bind here, but now the product size would be longer, right? And since most of the introns are pretty long as compared to exon, your PCR may not amplify these long introns, and because it's not optimized for that, so number of cycles, the how long is your amplification step in your PCR would not let the amplification of the long introns. But sometimes the introns are also small, or maybe your PCR conditions are optimized even for the longer introns. So which means that there is still a possibility that even these intron spanning primers can amplify the genomic DNA. Right now, this can be taken care at another level. For example, you can run the gel electrophoresis, and your cDNA, since it is smaller 100 base pair, if you run the gel, it will come somewhere here. Okay, it's running faster, right? So this, I'm running the gel in this direction. However, your genomic DNA would be somewhere up here, 10 kb, let's say, if the intron length was 10 kb, isn't it? So by looking at this difference, you can simply say that, okay, there was amplification of genomic DNA, and my product was also amplified, which is cDNA. This is cDNA. But you get to know that it's fine, but I have contamination of gDNA. Now, in one of the ways you can actually isolate this band from this gel and do your further experiment. That's one thing, but these are not recommended ways to get rid of gDNA, right? So if the gDNA is amplified by intron spanning primer, it can be seen in the gel. And if you do not see this, it means that this intron was sufficiently long that your PCR was not able to amplify, and then you only see your cDNA sample, cDNA band, right? Now, what is the way that, is there any way that I can get rid of genomic DNA possibility, okay, altogether? So these are binding, but can I come up with a way that these primers don't bind at all to genomic DNA? Okay. So there is a smart trick. So what you do is the following. So this is again your let's say genomic DNA is your gDNA. And this is your cDNA. And of course, you obtain this through genomic DNA through the process of reverse transcription, right? Now, what I'll do is the following. I will choose one of the primer, okay? I'm going to choose one of the primer which is spanning the junction, exon junction, and other primer from anywhere. It can be the second primer could be from here, or it can be from here. Now, if I take these two, the first, so this is 1, 2, 3, and if I take 1 and 3, this is not only the exon junction primer, it is also a intron spanning because there is another intron in between, right? So the idea is the following, that one of the primers should span the exon junction, right? So this is your exon junction. Now, what will happen if you have genomic DNA contamination in your sample? This exon junction is absent here. There is an interruption by a long intron. So this primer will never bind to the genomic DNA because it is actually spanning, the primer itself is spanning to 2 exon. Now that primer cannot span to 2 exon in the genomic DNA because the 2 exons are interrupted by a long intron. Okay, so it's no way that this primer will bind over here. So if you use this trick, and this is exon junction primers, so you will be able to get rid of genomic DNA. Now this is one of the exercise that I'm going to, one of the assignment that I will give you in writing. So you need to design primers by different methods: the ordinary primers, the intron spanning primers, and the exon junction primers. Right. So this was all for the day. In the next class, we will look at some more things which are associated with the experiments thing. We do some more analysis of the gene sequences to find out, uh, to actually optimize the expression level in our uh, during the cloning experiment. So when you clone a gene into E. coli, it may not express well, in a sense that expression in the terms of protein. Okay, so you have taken a human gene, let's say human insulin gene, and you put it in E. coli, and you learned multiple things. The first of all, uh, human genes are having introns. Okay, so basically your cloning should start from your mRNA to make cDNA and clone the cDNA of that gene into E. coli. The second problem is that E. coli is not expressing human gene properly, right? At the protein level, let's say the gene expression level at the mRNA level was fine, but that protein was not formed, and you want to make sufficient protein in order to purify that protein in sufficient amount to do enzyme assays, or purify the protein itself for structural determination and so on. So the reason is the tRNA pool, which is different in human and in E. coli. So you need to do something called codon optimization. Okay, we'll see in the next class how we actually do the codon optimization.